So, to make a complete glucose molecule, how many ATPs would you need?
Nine.
18, right? Because you have to turn this twice. 6 plus 3 is 9 times 2 is 18. Total, how many electrons are you going to need? 12. Exactly right. Boom. Photosynthesis. Okay, so does this kind of make sense, you guys? Where did we get our ATPs and our ADPHs?
From the light reaction.
From the light reactions, right? It's just from the light reactions. This stuff all comes over here to where the dark reactions are occurring. And we throw it all together with a bunch of carbon dioxide and viscose, and we make a G3P.
So basically, it has to go through the Calvin cycle twice in order to make a sugar.
Right, yeah, if we consider each turn of the Calvin cycle to have 3 carbon dioxides associated with it. So let's say that we're using just, and really, the Calvin cycle uses 1 carbon dioxide. I used 3 just for the sake of simplicity, right? So if we're using 1 carbon dioxide molecule, we take 6 turns, which should make some intuitive sense, right? Because basically we're taking a 1 carbon molecule and we're trying to make a 6 carbon molecule out of it.
Usually in nature it does one cycle.
Kind of. I mean, you have these reactions occurring simultaneously throughout the chloroplasts. So just how you want to look at it is really what it depends on.
So what about plants that don't take in sunlight? Like how do they?
Oh, that is a wonderful question. Like what? What doesn't take in sunlight? Don't they? So, almost all plants photosynthesize. There are some plants that live under the water that receive different wavelengths, and they'll have different pigments. They still do photosynthesis. They just use something other than chloroplast to be a photoreceptor. So like brown and red algae live so low that they're below the point at which blue light and red and yellow lights actually reach into the water column. And they're most receptive to green lights, so they'll photosynthesize with that. Yeah, Adam?
This might be a dumb question.
Probably not.
Mushrooms.
Oh, mushrooms, that's a great question. So mushrooms aren't actually plants. Mushrooms are
they're
fungi.
fungi?
Yeah.
Sorry.
So that's a different No, it's not a dumb question at all. In fact, there are some plants that act very much like fungi, and these are non-photosynthetic plants. There are parasitic plants. There's one called dodder that doesn't produce any chlorophylls at all. It's orange. And it has little roots called hostorial roots, and they burrow into a host plant, and they just absorb the photosynthate, the sugars that that host plant is producing. Yeah, Sophia.
So, since we're talking about like plants, what about what are those plants that like the Venus flytrap?
Oh, wonderful question.
Like, do they like they eat?
They eat bugs.
Yeah.
Yeah.
So
What's up with that?
I don't know.
Yeah. So, question. They're called carnivorous plants. These carnivorous plants, are they green? Have you guys ever seen a Venus flytrap? It's green, for sure. So do they photosynthesize? Absolutely. The question is, why are they also eating animals in addition to photosynthesizing, right? And the answer is because there are other nutrients that are important for plant growth other than just sugar. So typically, these carnivorous plants live in soils that are very nutrient poor, like bogs, for example, that don't have any nitrogen compounds. And so carnivorous plants are using animals to extract nitrogenous compounds from. Now, we've talked about 2 biological molecules that use nitrogens. And can you remember what they are? So I'll give you your options again. Sugars, proteins, nucleic acids, and fats. Proteins is one, for sure. And nucleic acids is the other, right? Because an important part of an amino acid, which is the monomer for proteins, is that nitrogen side group, right? The nitrogen attached to 2 hydrogens. So bog soils, they're typically glacial soils into which very few nitrogen compounds have been incorporated. So carnivorous plants are able to use animals to get their nitrogen compounds. And coincidentally, that's exactly how we do it. We have to eat animals or other nitrogen-rich plants like soy or corn or things like that in order to get those nitrogenous compounds. There is a ton of nitrogen in our atmosphere, but we can't convert nitrogen gas, N2, into some usable form. There are other organisms that do that. Great question.
I'm waiting for it, the carnivorous plants in there, like those plants that, what do they call? I don't remember what they're called, but they almost like a hole, and they just like take the sacs into it.
Yeah, so there are carnivorous plants in the tropics, and the tropics are another area that have really poor soils. Most of the plants, though, that you see living on the side of other plants are epiphytic. They're neither hurting nor are they benefiting the plants they're living on. But there are some carnivorous plants in the tropics, yeah. Other questions? Okay, let's go back to the PowerPoint presentation really quickly. And do our dark reactions. Okay. So in step one of the dark reactions, what we're doing is we're taking 3 carbon dioxides and we're attaching those 3 carbon dioxides to Rubisco. Now each Rubisco consists of 5 carbons, so across the Rubiscos, if we have 3 of them, there are 15 carbons, right? And we're attaching 3 carbon dioxides to it so that the intermediates are 6 carbon molecules, and we have 3 of those. Now those 6 carbon molecules are short-lived. They split into 6 3-carbon molecules, which ultimately become G3P. Okay, and so that was the fixation phase, right? In the reduction phase, what we do is we take those 6 carbon molecules, split them, attach some phosphates to them, and reduce them by attaching electrons into G3P. And we create 6 G3P molecules. So we took our 18 carbons from those 6 carbon intermediates and convert them into 6 3-carbon molecules. Now, again, this accounting ought to make some intuitive sense, right? We're pulling 1 G3P out to make sugar, 3 carbons, and that's how many carbons we brought into the system, 3 carbon dioxides. So we're pulling one of those out that's ultimately going to become sugar. The remaining 5, and we know that there are 15 carbons, are reclaimed back into Rubisco. And we require 3 more ATPs for that. And that is essentially the dark reactions. What's that?
It's a lot simpler to diagram. than
Yeah,
diagram.
it is. Not nearly as fun. Okay. So this is a more expansive diagram of the complete dark reactions. So the actual product that we receive from the dark reactions during the reduction phase is 1G3P. And you just need to think about G3P as a sugar intermediate. It is a molecule that ultimately will become a 6-carbon sugar. So again, we have our 3 Rubiscos that are attached to 3 carbon dioxides to make some 6-carbon intermediate. That's the fixation phase. Then during the reduction phase, we take that 6-carbon intermediate and we attach phosphates to it and we attach electrons to it until ultimately we get 6 G3Ps, right? We pull one out, the remaining 5 go into the reduction phase. The question is, what happens to our ADP and our NADP+? And the answer is, those carrier molecules are recycled back to the light reactions. So when ATP is spent, it becomes an ADP, it returns back to where the light reactions are occurring so that it can be phosphorylated again and go back to where the dark reactions are occurring. And the same thing with NADPH. When it loses its electron, then it returns back to where the light reactions are occurring to pick up more electrons. Which is important, right? Because you don't want to have to remake NADP and NADP plus every time this reaction occurs, because it's occurring with some frequency.
So basically what it is it goes back to the light reaction design.
I'm sorry?
So what an ADP and NADP plus lose in the dark reaction and go back to the light reaction design.
To pick them back up, yeah? And they're just constantly shuttling either phosphates or electrons through the entire photosynthesis process. Okay. I'm going to skip that. So let's end here, because this next topic is something I would like a fresh mind for, and it's called photorespiration. Do you guys have any questions so far about the dark and the light reactions? Is there anything other than that diagram of the light reactions that you would very much like to see on the next exam?
The dark reaction.
What's that?
The diagram, do it on three.
Yes.
A diagram of the dark reactions?
Yeah.
Diagram.
So how about questions like, which of the following is the stage in the dark reactions in which carbon is attached to Rubisco?
That's fixation.
It's fixation, yeah. So expect questions like that as well, I guess. Yep, Linda.
So basically, the dark reaction, that's the Calvin cycle.
Yep. Okay, great. I will see you guys next week. Have a great weekend.